Hi Year 12s, today we're going to learn about respiration. So first of all, let's take a look at what our study design wants us to know. Today we're going to focus on the main inputs, outputs and locations of glycolysis, Krebs cycle and electron transport chain. We also are going to look at the ATP yield of each of those different stages. We'll look at the location, inputs, and difference in the outputs of anaerobic fermentation in animals and yeast. In our new study design, you might have learnt about anaerobic respiration in the past. They've simply changed the word from respiration to fermentation in the new study design, and again helps us, I suppose, to have a clearer distinction between aerobic respiration, which requires oxygen, and anaerobic fermentation, which does not require oxygen. The purpose of cellular respiration is to transfer chemical energy stored in glucose to chemical energy of ATP for the use of our cells. Before we go any further, it's important for us just to stop for a second and consider what is ATP. So ATP stands for adenosine triphosphate, and it's a source of energy that keeps us going. It powers our biochemical reactions in our bodies. So you can see a little diagram just here where we can see some phosphate groups, and we should be able to count 3 of them: 1 phosphate, 2 phosphate, 3 phosphates attached to a ribose sugar and an adenine group. When we release energy, ATP breaks down into a molecule called ADP, or adenosine diphosphate. So basically, when we're breaking down ATP, we go from having 3 phosphate groups to 2 phosphate groups. And the breaking off of that single phosphate group, which you might see sometimes denoted as Pi, releases the energy that we need in order to power reactions in our bodies. Let's now take a look at the mitochondria. The mitochondria is a cellular organelle. It's found in all eukaryotic cells, and our mitochondria is responsible for producing, or is the site of, aerobic cellular respiration. So the mitochondria has a double membrane, and that's because it's evolved as a result of a process known as endocytosis. Mitochondria used to just exist as a single structure a very, very, very long time ago, which was engulfed by other organisms over time, which is why it now has a double membrane. And the double membrane serves a great purpose for us when we're doing respiration. So we can see an outer membrane on the outside, obviously, and then a really folded inner membrane inside. We've got a mitochondrial matrix, which I call as like the mitochondria juice. And then we've got little proteins located on the edge of that inner membrane, which are called the cristae. We'll have a look at what these structures are responsible for doing and helping us with respiration shortly. So there are 2 types of respiration. There's aerobic respiration and anaerobic respiration, or anaerobic fermentation, which we call it in the new study design. We need to know in Year 12 biology how to write a chemical equation for respiration. So we need to know that glucose combines with oxygen to form carbon dioxide and water, and in doing so releases ATP. We know that glycolysis, the first stage of respiration, happens in our cytosol, and if there's oxygen present, we do aerobic respiration after that, where we see the Krebs cycle and the electron transport chain happen. If oxygen is not present, we do anaerobic fermentation, which produces lactic acid in animals and alcohol, or ethanol we sometimes say, in yeast and plants. Let's take a look at anaerobic fermentation. This type of respiration, as I said before, occurs when there's insufficient oxygen supply in a cell, or when the cell is in need of very quick energy for short term. It happens in a process called glycolysis, which also occurs in aerobic respiration. So let's now have a look at that process. So in glycolysis, we start in the cytosol. So if we imagine a cell, our cytosol is the cell's juice, and inside the cytosol we contain lots of different organelles. Okay, one of them is the mitochondria, which we'll talk about a bit later. When we do this process of glycolysis, one single glucose molecule is broken down into 2 pyruvate molecules, which is a 3-carbon molecule. We produce 2 molecules of ATP, and hydrogen gets taken from the glucose and transferred to an electron acceptor molecule that we call NAD. And in doing that, NAD ends up making a molecule NADH. This is a little summary diagram of this process. So again, we can see our glucose, 6-carbon molecule, being broken down to pyruvate, or 2 pyruvate molecules. And in doing that process, we see the production of some ATP and some hydrogen being accepted by our carrier molecule NAD. The outputs of anaerobic fermentation are different in animals and in plants or yeast. So in animals, when we've converted that pyruvate and got the energy that we need as a result of that process, we produce lactic acid and water as byproducts. Okay, so lactic acid and water are the outputs of anaerobic fermentation in animals. In plants, yeast or bacteria, this process of anaerobic fermentation leads to the production of ethanol, which is a type of alcohol, and carbon dioxide in plants, yeast, bacteria. Okay, so it's a little bit different. We get different byproducts at the end, okay, but the process to get them is the same. This here is a little diagram which shows that information visually. So we can see glucose being broken down into the pyruvate. It figures out that there's no oxygen available. So what happens is that pyruvate or pyruvic acid, which it can also be called, is broken down into lactic acid in animals and ethanol and carbon dioxide in plants.